Who knows what is the difference between supervised versus unsupervised learning? Good. Okay, so this talk for you guys first will be mostly about unsupervised learning and how it could actually help with complexity in science. For those that don't know what this means, hopefully at the end, just a little bit will make sense. Okay, so the talk's title is Tokenizing the Language of Complexity in Science, basically about coarse-graining. I try to stay as close as I can to the abstract that I gave you with one or two small omissions for examples, because I'm trying to keep it within time. Okay, so first, who am I? You probably don't know me because I'm on that side. In fact, I'm even further. My office is in biology. I don't have a physics office. But theoretical and computational physics was my training. And then I joined Slack, not the app, but the lab. As a postdoc, came to Singapore. 2016 was when I started the lab. So who are we? This is the group part. Everything that you see here is done by other people, almost. So these are the research fellows, PhD students, and master students. And the takeaway, if there is one for today, is we need to think about training bilinguals, people that know science and AI. And we've been fumbling through trying to do that since 2016. My own personal training came from doing statistical learning, which is a noisy form of machine learning for X-ray diffraction imaging. Usually when I show up this slide, I also use this as an opportunity to tell people that as young people doing a lot of different research projects, it's good to have a visual business card that show the stuff that you do, right, and summarize that. So that's mine when I was a postdoc. Because we do it for X-rays at very low cross sections of scattering, it became useful when the signal to noise was way higher, like in electron microscopy when it came to Singapore. And so then we could use it for another field of imaging where the signal to noise was way higher. We try to push it down low with the methods that we developed for X-rays. Okay. Yes. A lot of people ate food because papers, blah, blah, blah. Thank you. More importantly is thank you, money people, for supporting the journey. There are 5 AI-generated images. If you get bored or tired, you could try to spot them. Okay. All right. So this is the starting point, which has become the starting point for us. Okay. A lot of the data and the people that we work with, typically they come to us because they have complex data. And so over time, we have to start to understand, like, what does it mean to understand complexity became a more and more central theme within our group. Okay? And here is when I move into the analogy. If you hear another person speaking a different language from what you're used to, it sounds pretty complex. It sounds very confusing. But you also use a language that is complex and confusing to another person. So language, however, comes naturally to all of us, and we're all experts in at least one language. So the question is, can language be a possible model for complexity? This is unproven. It's extremely hypothetical, and we're pursuing this line of thought. Okay, so what is language? If you break it down in a very too reductionistic way, you probably know these terms because of language models these days. It's made of tokens, grammar, and context. Okay, I hope maybe 3 years ago I had to explain it, but probably this 2025 you don't really need. I'll explain a little bit more. When I was a grad student, we used to use this concept of a bag of words, which is basically a bag of context. A bag of tokens. Okay, so here's a bag of words. Does anybody know what is the article for which this bag of words came from? It could be somebody's report. It's Singapore, right? Because you can see the shape of Singapore. That's the other hint. If I were a magician, that's what I would do. I would change the shape to Malaysia or something, just to throw you off. Yes, this is the wiki article for Singapore, but just knowing the bag of words doesn't really get you the full context. Also, the grammar is lost. And we used to use this a lot for language model detection. So language isn't just a bag of words. See, this will be pretty surprising for me. So how do we understand complexity as science? Okay, so I'm going to switch gears a little bit to complexity in science. Okay, so that was language. And I'm going to talk about complexity in science for a little bit, and hopefully you will see the confluence with two of them. So science is also very complex, but the way we understand science it turns out we were taught in a very reductionistic method. Okay, and in physics, we like to call it a coarse-graining. And the main idea behind coarse-graining is this very simple rephrasing that less is more. Because you simplify things, you can scale up to larger length and time scales. Okay, so coarse-graining allows us to span multiple lengths. So if you look at all the textbooks that you have on your shelf, you pull out any one of them, you pull out another one, and you try to connect the two of them. If they're at different time or length scales, sort of different domains, there isn't another book that tells you how to bridge them directly sometimes. Famous quantum gravity, right? So that's one example, but there's actually a lot because our domain knowledge and our models basically work very well for limited length and time scales. And the reason that works is because we have to reduce it to that level. Okay. So this is the example I like to give because this is a colloquium. So this is, sorry if this is a lot. I know you guys are very smart. And you're also physics smart. That's a different kind of smart. Okay. But so bear with me. I usually use this for people like me. Okay. So this is an example of reductionism for something that you and I can relate to. So we are organisms, but if we reduce it down a little bit, we're basically a bag of interacting organs, right? And so you have something wrong with this part, you look for this part doctor, not this part doctor, whatever that part is. But each of the organs has another hierarchy, which are made of tissues. So when you do a histology, you basically do sections of that part of the tissue doesn't, there's some problems. And tissues are made of cells, and then cells are made of proteins and other things. So you see this explosion of complexity as you go down. And the reason we see this is because our instrumentation got more precise, more accurate, and high throughput. So this is partially our fault for creating magnificent instrumentation. Now we can see all this complexity. And the following question is, how do you bring it all back up? Now that you can see all the pieces, how do you bring this all back up to create that organism? And this we don't know how to do very well. Just with proteins in a beaker, shake, 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 a person doesn't appear, right? And the reason why this is difficult is because more is different. Now, here's number 2. Does anybody know what said this? Ooh, very good. Nobody. Wait, what? Okay, I knew it. I can do this in other places. Yes, okay. Shh. Okay. More is different. What does that mean? Okay, 2 equations. You're familiar with this, right? What do these 2 equations have in common? Anyone? Let's start saying all kinds of stuff. Heat. Anything else?
The involve a product.
Okay, the form of a product, yes. This is intentionally picked to be as different as possible that I can think of. I'll explain a little bit, okay? I'll explain using nucleation, something that I have thought about quite a little bit. If you've thought about nucleation theory, it's to predict when a crystal will grow out of a melt. So the idea in classical nucleation theory is that there's a critical size, a group of atoms that have just spontaneously come together to create a single crystalline unit. And if they reach a certain size, then it would only grow. If it's smaller than that size, it will shrink and disappear. Okay, so this is the classical nucleation theory. And so if you think about this, any bigger, it will be stable, it will grow. Any smaller, unstable, it goes away. Okay? How do we think about this is very simply that there's a surface energy cost and there's a bulk energy saving. So this is plus, this is a cost against the rest of the not crystalline part. And what we do here, because we know it's proportional to, is we rewrite this in the form of specific coefficients. So this is the surface energy, sigma, and then that's the bulk savings. These numbers are coarse-grained. Typically, if you have a molecular dynamics or DFT simulation, you find ways of jiggling around and measuring these things in a coarse-grained way. Okay, so these are the coarse-grained parameters. Okay, and so if the surface energy is bigger than the bulk, then it shrinks. If the bulk is larger than the surface, it grows. Very simple. So this is the guy that said it, Phil Anderson. More is different. He wrote a very nice paper in 1972 that continues to be cited heavily. And the most recent papers would be 30 years after the more is different. We're still struggling to understand what is different. If you rephrase it a little bit, it will be the sum is more than its parts. Okay, so this is related to emergence, weak and strong emergence if you're into philosophical bits. Okay. So those equations, these two parameters are emergent descriptions. These are coarse-grained parameters, right? These are static coefficient and temperature. These do not exist for single featureless particles. Temperature doesn't seem to make that much sense for a single featureless point particle. It's only in aggregate that the sum happens, then the temperature can be well defined. Because you can always move to the center of mass frame of that one particle, and then the speed is gone, so kinetic energy is gone for a point particle with no features. Same thing for static coefficient. We don't really know how to do this from first principles, except we jiggle these molecular dynamics, and then we find out what it would have been to fit this formula. So these are emergent properties that allows us to coarse grain. So we can do friction in an engineering sense without really caring so much about the atomistic or even the quantum mechanical details. And this allows us to scale in time and length. Okay, so we can reduce things into a lot of different complexities because of instrumentation, but to build it back up, I claim that we have to coarse grain. And this is very related to all the coarse graining models that you're seeing typically if you're into the, the computational materials sort of world. Okay, so this is the link, hopefully. So if you think of tokens as building block for language, what is the building blocks for complexity? Okay, I'm going to introduce the idea of motifs because motifs are easy for my brain. It's very visual. And if you remember anything about motifs are little bits of repeating tiles. Like if you go to some, I don't know, has anybody been to nice religious places with nice tiles? I see a few people nodding. Okay, you know what I mean. It's a certain arrangement of tiles that keep repeating, but in a sort of suggestive, maybe not completely periodic sense. So I'm going to show you, yes?
Can you elaborate how different this is from a pattern? Since we're calling it a pattern?
It's the same. I'm just making up words. The pattern, you can think of, so.
Pattern.
Sure. Then, and there's, if you, later on, I'm going to tell you about patterns between patterns, and I call it a hierarchy of motifs. So you can call that a pattern too. It's whatever level you want to coarse grain. Okay. So none of these definitions are well defined, but I try to. Okay. Yes. So the difference now is that machine learning can help us find, using unsupervised learning, new motifs, things that are not easy for us to find if we just did it with pure brain power and observation. Okay. And what it will do essentially is helping us pay attention to which details to pay attention to and which ones to ignore. And the ones you ignore, basically coarse grain away. Okay, very simple idea. Okay, this example was done by Jiadong when he was a PhD student with Steve Pennycook at Materials Science. He's now a Schmidt Fellow in the group. And we're also using some of this work. I have to acknowledge all the people, right? These are the people that we work with. Okay. So Goki, Suing, you might know them. And Michael Bossman, he's an electron microscopist over at Material Science. Here's the question. Here's a sample, molybdenum diselenide. They're looking for these tiny point defects in there. So molybdenum diselenide, if you know, it's a triple layer. It's like a sandwich. So peanut butter in the middle, right? That's molybdenum. The sandwiches above and below are two layers of selenium above and below, right? But they're arranged in a hexagonal way. So you have two selenium on top, and then next to it would be a molybdenum in the middle. That's the peanut butter. Okay. So they're looking for these single point defects where of the two selenium, one of them is missing. And these have some properties that give it single photon emission. So where are the defects? And you see it. People used to do this by eye. So I can flip back. Right? Now you can see them. Well, actually, it usually depends on the projector. Okay. So how do you detect these little motifs, these tiny little pieces, the defects that appear everywhere? So I'm going to describe to you how I do it here, and hopefully we can generalize this more across different examples. The way to do it is to take every single patch and then extract them out, and you have a pile of these patches, each centered on an atomic column. Okay. Now, this could be a molybdenum. This could be a selenium. I don't know, because I'm just looking at some column. Remember, they're all offset from each other. Then try to do this. Does anybody know what I could do to do this?
Clustering.
clustering. That's a good one to start with. What?
Means.
Clustering.
Okay, so that's for the coloration, but there's another step here, which is dimension reduction. Yes, PCA, right? So the question mark here is actually the two of these things combined. A lot of very powerful techniques. Who has heard of PCA? Wow, this is a good crowd. Okay, who has heard of TSNE? TSNE. Oh, not so bad, not so bad, not so bad. UMAP. UMAP is usually, okay, so there's a whole bunch of generations of tools that go past PCA. PCA is like if we took the 3D positions of all of us in this plane, took a picture in 3D if we could, and rotated askew, right? And somebody says, this is not level. Let me level this with a simple rotation so that the plane that we're all sitting on is on XY. That's what PCA is, just a simple rotation. But when we live in high dimensions and we're all over the place, there's another way, which is a non-linear, non-rotating form that brings us down to a two-dimensional plane that keeps the pairwise distance between all of us in the high dimensional space the same as it would have been if we were in 2D. So these are the more powerful nonlinear dimensionality reduction methods. I'm glossing over a lot of key details, but this is roughly what it is, okay? So you can do that, and that's what we have been teaching a lot of. This is very simple, like unsupervised learning. But you see that there are different groups, and that's where I think the comment about clustering comes in. One of these groups, each dot there represents a particular patch. So there's a whole group of patches that actually correspond to selenium-centered crystals. And then there's another one that is molybdenum-centered vacancy. The molybdenum has 3 nearest neighbors, and they could have vacancies at 3 different places. That's why there's 3 different dots, right? 3 different places. And so the question, and then there's selenium-centered vacancies. You're on a vacancy, you're centered there, and then what's around you? Okay, so the question now here is, do you pay attention to the variations within that group or not? If you choose not to, if you understand the variations within it, and you choose not to pay attention to it because it's not important for you, you can coarse grain it away. And so this is a very nice visual example of what coarse graining actually means. You see the degrees of freedom within class and between classes, and then you make the decision whether to pay attention or not. Okay, so now that we identified, okay, by the way, one of these clusters, you can call it a motif if you want. So it makes sense because it's a group of patterns that are similar to each other more than another group of patterns. Okay, if you give me that liberty to do this, then we can start asking the question: what is the this is a bad animation. Sorry. what is the relationship between motifs? Right? What does that mean? Motif hierarchy. And in the language model, this is token grammar. So if you find a motif here, what's the probability of you finding another motif next to it? If I say the words the apple, what happens next, right? What is the apple token grammar? The way to do this spatially is quite simple. You just look at all the different examples that you had, and you replace all the different columns with its cluster ID, right? This is just a simple motif, blue type motif, orange type motif, whatever it is. Then you ask the question, if you're next to a blue type, are you also likely to be next to a whatever purple type and so on and so forth? Then you build basically a hierarchy of all possible combinations of these things together. That is what I mean by a motif hierarchy. That sounds a little bit like a token grammar. Okay. All right. So from some of the examples that we've seen that were published on, there are this is straightforward, right? This is the check that makes sense. There's discrete motifs, simple words, with simple hierarchies, very simple grammar. They don't interact very much, very low density. Then there's the higher density case where discrete motifs can come with different tiling forms, so the hierarchy becomes much more complicated. And then there's the worst kind, which are continuous. This is like a person that slurs. You can't really hear their words properly, right? So these are the continuous ones, and it would manifest as a sort of like a manifold. There's some topological stuff that we don't have to extract from. Okay. So let's see. Right. So if you think, oh yes?
This defect picture you showed.
Yes.
Aren't those defects random? So if you find some motif or pattern there, isn't there some kind of hallucination? I mean, we can also see pattern in random things.
Okay, so for now, all of these, yes, that's true. That's absolutely true. So for example, we can see constellations. That's a famous example. Constellations are made of all these galaxies that you think they're like in, but in fact, they're hundreds or millions of light years away from each other. They're not actually close. Yes, these are just patterns for the sake of finding patterns for now. The more important thing is to see whether you can find a mechanistic reason for them to have the pattern or not, whether it's a simple correlation or there's a mechanism behind it. And if I jump the gun all the way to the end of the talk, is to say machine learning is good for finding correlations, but correlations are not causative. The mechanisms behind it is what we need to go in and try to figure out. Sometimes the mechanisms are purely absent. Sometimes it's something that we have not thought about. Okay, so erase all of that. Let's go back to you didn't hear that. I'm gonna continue with the spiel. Good question. Okay, so now you see the motif. Now you see the hierarchy. Let me go back to the language model again. I'm learning a language that I don't know. Okay, I don't know a lot of languages. Easy to pick one. Let's say Italian. Who speaks Italian here? Maybe some people. Okay. Two scary people speak. Okay, so if I learn Italian, I learn the words, I learn the grammar. What do I do next? I start talking to a person that speaks Italian and practice and decide that no, you're saying this wrong. So through that practice, I figure out how to tweak my internal model. Maybe it's a grammar thing, maybe it's a context thing, maybe, you try it out, right? So once you have the grammar, the natural thing is to say whether you can create passages, right? So this is the motifs, right? This is the generator of complexity. I'll show you an example of this. This is a piezoelectric. Somebody published this, our collaborators 2022. So piezoelectric, if you remember, is the very annoying bit of LED light at the bottom of children's shoes. Or you use it also for experiments, right? So in this case, there's a sodium potassium niobate where a lot of the piezoelectric effect happens at the grain boundaries. So there's accumulation of charge that happens around these strain places. And if you apply an electric field, it will the charges will move away and will cause a mechanical change in the strain that is accumulated there. Okay, so they know this, and it's a very giant effect. So all of this effect, they have been modeled as just along the grain boundary. But then they took that image of this thing. This is very nice, right? Periodic, nice and straight, grain boundary. What does the grain boundary look like? Yeah, it's snaky everywhere. It's non-periodic. Okay, so the question is, can we describe this? That's the first order goal. This is complexity. So is this truly random? Is this a random walk? If you know anything about random walks, you would immediately say this is not a random walk. Random walks are like clouds. They're scale free. If I show you a picture of a cloud with no other reference next to it, you cannot tell me whether this came from a steam kettle or it came from the cloud or like in the sky. It's scale free. So this doesn't look like any random walk that I know of. There's some repulsion between it. So the question is, can we write this as some sort of generator? Okay, how do we do this? Okay, you should slowly get into the groove. The same thing, get the tokens, get the grammar, but what does this look like? So these things are actually made of these niobium with oxygens around it. And then they can't, in each of these are actually octahedra with oxygens you can't really see. Right? But they can't in a little bit to create a little hole in the middle. And that's all important for the boundaries. So there's two types, triangular, hexagons, okay? You coarse grain all of that away, I'm hiding some of the details, the same stuff you saw earlier. We can coarse grain out of the way into triangles and hexagons. And triangles and hexagons can only pair up in a certain way, okay? Because of geometry. And then once you have that, then you can ask, if I have two of these shapes, what is the probability of me adding a third shape? In the language model, it would be if I have two tokens, what's the likelihood of the third word, right? CQ, oh no, wait, that doesn't work. Center for quantum something. You know what I mean? So you can sort of guess what the third word is. CQ could be a whole bunch of stuff. So that is the two motif, the three motif. Each of the rows represents one of the possible two-dimensional shapes. The columns represents what is the next one that will come. Okay? So this is already a very simple grammar, and it's only up to three motifs. And we looked through all the images that you saw on this weird, random walkie thing, and we accumulated the grammar that way. Imagining as you move along on these three motif shapes, picking up the grammar and averaging it over to create this matrix. So this is a possible grammar for the motifs. And then we can start creating weird passages. These are all made up, fake. Because once you have that, you can say, well, I know these two make a third one. Well, I shift one again. And then now I look at the last two and I ask, what should the next one be? And just keep adding words. This is how GPTs work, by the way. Okay. No surprise. You just add on the next thing, giving a longer context. Except for a lot of GPTs, the context window, like GPT-4, the context window, as far as I remember, this morning, my chat with GPT, it told me 12,000 token is the sequence line. So it looks at 12,000 tokens and it says, what's the next one? Okay, this is just 2 and then the next. So we can start generating all of these things, but do we know it's right? So just to set the tone, we get that. We tokenize it. That's the tokenized part. Because I would say motifize, to make a motif of something, but that's not a real word. So I use tokenize. We create the grammar, which is the transition matrix, generate samples, and then we test. We go into the actual material and say, well, let's speak this language and see if it matches with what people say. Okay. And then we ask the question, is more really different? Because if it's really a random walk, right? It's like die roll. If it's a real die roll that's really random and fair, I should be able to just tell you the probability of a die roll, and you should be able to guess with high precision, large batch of die rolls, what the average would be. But if there is a variation between the two, then you know there's some physics that you're not really sort of accounting for. So all of these probabilities that we calculated in the matrix are just from near stereo correlations, seeing 3 words as you go, 3 tokens as we go. Then we ask a different question. If I say you these 2 words, what is the 30th word down, 20th word down? We test that. So in experiments and simulations, the correlations between those types of tokens match up pretty well. So what does that mean? This means that this thing's really quite random. After the 3 tokens, it's pretty much random. Except past that point, it sucks again. So what's happening past that point is we try to look for the scale, the large scale sort of critical exponents for like, I think we also looked at, so like the Hausdorff fractal dimension and other types of morphological constants, they don't match up. And the reason they don't match up is because we didn't take into account the domain-domain interaction and the wall-wall interaction terms. So then we baked that into the whole model. This is basically saying, I'm trying to write a text about physics. Can you come out with the right words? So the context has to be important. Then you use the generator to create the small passages inside. Now it's a passage about biology, write the stuff inside. So this hybrid approach of giving the context that we have to give, then this is the physical context they've given. And then the words that go in, this is what basically we have to do for everything to match up, okay? I hope you can trust me. Okay, now I'm gonna go really crazy. Okay, that was space. We do space and time. Ooh, okay. So this is the bio part of me showing. This is recorded. So I'll say for the sake of the recording, I know some biology. Good, right? Okay. So tokenizing self-organized cells. Okay, what does this look like? And simple interactions lead to complex collective behavior. So does anybody know what this is? Cells. Bio cells. This is from slime mold. A specific slime mold called Dictyostelium discoideum. This is the only slime mold that I know. And they do this very interesting thing. So you and I are famously multicellular organisms. Okay? You're not a single cell, you're many cells together. This cell is a slime mold cell that does its own thing. Just, goes around, looks for food, blah, blah, blah. When you starve it, they release a signal. And the signal says, I'm starving. And the other guy says, you're starving too? And then they start having collective behavior. And they start swarming together to form a mound. The mound, there will be an internal little cyclone within the mound. All of these were single cell organisms that don't care for each other before. But when they're starved, they'll form these little mounds. And the mounds would start differentiating. This is a time series snapshot of a few different things put together. And then they will start creating a stalk. And at the very top of this, and they'll move around, look for food, because you're starving, maybe we can move together, we can find food better, right? And then at some point, the stalk would keep moving and then concentrate at the very top, a high pressure globe of DNA enriched set of cells. And that would keep pushing to the very, very top. And because of the high pressure, it would pop randomly in some direction and then shoot all the DNA material in some other place, hoping to find food and establish a new colony there. And this is the stuff that physicists love to study. We call it dicty. Okay, so there was a model that was created by this gentleman, Tetsuya, and he basically created a simulation of this that sort of mimics many of these different types of dynamics that we've seen in the cell, but in two dimensions. And my PhD student, Ying Chen, who was Professor Lim Hock's FYP student, that's why they can hear that. He started thinking about this, whether you can find spatial temporal motifs from this whole thing, because it's dynamics. Okay, so the model that this guy had was quite simple. It's basically a little, if you know anything about active matter, it's things that cannot stop, right? It will always move based on a compass that it has. So every cell is sort of modeled with a little compass on it. And there's two parameters. Okay, and it will move in the next time step according to where the compass is pointing. Okay, no explicit forces, just accelerations and change in accelerations based, no Hamiltonian. No Hamiltonian. Easy, right? To explain to you guys. Okay. One parameter controls whether or not one of these compass would align to its neighbor's compass, like birds flocking together with a similar path. This is called the contact following. And then there's another parameter, which is if they come close enough together, do they tend to stick and move forward, or do they repel? You can think of this as inelastic versus elastic collisions. Physicists talk for this, okay? And with just these two parameters changed, scalar, okay, you can have a whole bunch of different types of behavior. You can have rings, you can have like motile aggregates, or you can things that don't move, you can things that swirl around. And this was published already by Tetsuya in a very sort of thought-provoking PRL back in 2020. So he did that. This is from his paper. Every single one of that little pixel is a movie of this thing. I will show you 8 of these movies in a little bit. He watched 861 movies and classified all these things. Why? That alone should award him a PRL and maybe 2 more. But we were wondering, can we do something about this? Okay, 8 of these movies. Okay, so I don't remember where I picked it from. Each movie has a different set of parameters. And ready to go? Spatial, temporal, unstable, stuff like that. So the goal for us was that little boneheaded idea of spatial motifs that we had with electron microscopy and materials. Would it work here? How? And there's also the dimensions of time. See, some of them move around, some of them just like huddle there and do nothing. Most of these are unstable. Okay, so in this case, how do you define motifs? Well, let's philosophically, like, sort of debate a little bit about this, right? Motifs should be persistent and prevalent. So something that stays around enough for you to call it a pattern. But it should also be prevalent. It should be in many different places. So in that phase diagram, okay, wait, sorry. We had a very simple idea for motifs. So it's either 5 around the center, so 5 nearest neighbors. So 6 things together, the 5th one in the middle, 6th one in the middle. 6 nearest neighbor or 7 nearest neighbor. This is a very typical situation because you can't pack everything too densely. There's 8 and 9 and so on and so forth, but they occur less commonly. But you can see that they're persistent. The 6 nearest neighbors and the 4, 5 and the 7, they have a pretty high persistence, right? In this 2 dimensions of all the movies that you saw. They're also quite prevalent. They're around quite a bit. So that makes it a good candidate for a motif. Okay. Then what we do? Well, we pretend to know what we're doing, and then we try to build the motif and the motif hierarchy from here. But each time you look at the motif around it, you need to find certain things to pay attention to. So who cares if it's 5 nearest neighbor? What do you care about it? Do you care about its density, its velocity, its curl, or its divergence of the cells around it moving? So you featureize it appropriately, and then everything else is what you said earlier. Okay, we can now create some sort of dimensionality and clustering to find whether there are these spatial temporal motifs. So these are basically classifying movies into descriptions. If there's a chatbot that is related to Netflix and it says, I want to look for, okay, this is a fun one. A lot of you guys are pretty young. I want to look for a movie with an old man with a gray hat that walks around with a scary stick and 4 little short people also running around it. And they were trying to find a volcano. What movie is this? Yes. Well, young people might not know this, right? But if I can describe the right words for this, this is what we're trying to do in this case. Right? Can we summarize all the statistics of all the things moving around into just 72 numbers for a single movie and cluster them. So I'm not going to get into details, but you can, okay? So you can summarize in the 72 dimensions so you have the coarse grain in time in a way that allows you to do that. Okay, so machine learning can do this. You can either use a neural network, but this is neural network free. And then you can cluster. Now, what we found is that these groups of movies are different. The disordered ones are the simplest. The black lines are the one that Tetsuya drew initially. So there's some matches and there's also some mismatches. And we're trying to now figure out whether or not there's actually new phases or there's some of the phases that he saw were actually pseudo phases. But this, we think, is a better way of actually finding spatial temporal motifs. There are no grammar here yet. We're trying to scale up. But this is just spatial temporal motifs. Oh, yes. So I apologize. This talk has suddenly been interrupted by a conversation that I had with Alex. It's just like a lot of people do imaging, so I should talk about imaging. So here's a different talk about imaging. I don't know how to do this transition. I'm just going to do it. But I will show you some of the techniques that we're now using. Basically, neural networks are powerful, but they can also be taught some of the prior knowledge in techniques that we have. Okay. So there's this concept, idea that I have been promoting called computational lenses. So the idea is, you know, physical lens, like this is a physical lens, or I can't see. Okay. But this physical lens allows me to see. Now the question is if you know how image is formed, right, through this lens, can you create a computational version of it? So you just have a detector and you detect wherever it is, and then use the fact that you know the image formation to create a computational version of a physical lens that you cannot make. The point is not to make a physical lens that you can make, it's to make one that you cannot make. Okay. All right, so we'll see some examples. I don't know why I put this up. Okay, so can machine learning make computational lenses? The answer should be this. I'll introduce this idea to you. How about 3D metrology? I'll show you an image. Right? I would like you close one of your eyes, you can, or just like do this, okay? Can you see where the flowers are far or close? You still have depth perception, right? So this whole thing about you need 2 eyes for depth perception is not really true, right? Oh, wait, this is wrong. This is Deepan. Deepan did the work. Sorry. Oh man, I gotta erase this from YouTube. All this work that you're seeing came from Deepan. I should have looked at this. Okay, Deepan was the big driver of this. When nobody believed that it would work, he just kept pushing. Okay, yeah, this is the one. Okay. Okay, sorry. So you were like, why do I look at this guy with a single eye? Okay. So near and far, you can tell with single monocular vision. There's depth perception. Okay. The other thing that you know about is, okay, I'm gonna stand right here. What you're seeing of me is a 2-dimensional image in the back of your retina. But you have a conception of me as 3D. I didn't turn all the way around at the top. How do you know I'm not 3D? Well, this is what we're doing to get 3D tomography for most of our instrumentation. We turn it around all the time, right? But why can't we have our version of 3D conceptualization with just, you know, what we're imagining is filling the blanks for what we know should be a person, what should be the back. You can imagine what's at the back of my head. Right? It's this hair. Soon it'll be the absence of it. So, but you can imagine that without me doing that. You can imagine apples in your head. All kinds of details. You can do that because you're interpolating. You've seen apples, you've seen the back of heads before. Our 3D instrumentation don't have that capability. Okay, so I'll show you an example of electron microscopy, very simple optics where you can actually do this if you understand the image formation mechanism. This is a very old school electron microscope. Nobody uses this sort of stuff anymore. It's now much bigger and much more expensive. But the image formation is still the same. As a plane wave of electrons, typically because it's monochromatic, go through your sample. I'm going to arrange 2 blocks of sample. A thicker one on the left that's further away and a thinner one on the right. Okay. So, you know, electrons are strongly interacting. So that means the thicker one would absorb more. The electrons that make it through will be fewer. So it goes through a lens. So that's why it looks darker, right? Fewer electrons made it through, more electrons made it through there. But now you look carefully. You take little tiny patches of the same image and you do a Fourier transform and you'll find different ring features. And what's happening is the one that's further away is more defocused. And so their defocused rings, which we know very well from image formation, the contrast is different. This very simple clue was the clue you used. See whether an object, the sakura flowers, are further or closer. It's how far defocused it is. We have it in our brain because of training that we've done with our brains when we were little all the way to when we were older. This we know the physics of. So the question is, can you use this, right? And say, okay, I know that part, P1, is closer to me because the defocus ring is a certain way. I also know it's thinner because it's lighter. Can I then place little objects in there, go to that position where I think the defocus is, and pop out the thickness that I expect? And repeat, repeat, repeat. So you can get a three-dimensional object from this. Very simple idea. Okay, so it's published, so I guess it works to some extent. Here are two different things, okay? Do you know what these are? The silicon nitride channel and a pit. And this is a single TEM image. This is experimental, so it's not made up, as far as I know. So we do the same technique, and you actually see that one of them actually has a pit feature, and the other one has a blowout feature. It's actually a hole that the electron moved through. And we know that this is not wrong because when we do this correctly, there's a flat underside to that thing. If it screws up, you won't get a flat thing at the bottom. And also for this one, usually when you mill through, it blows out because the debris sort of, the debris catastrophically explodes out. Okay, this is YouTube. I can't say more. Blow out. Okay, so this is the work that was done. Okay, now where does the neural network come in? Because all you have seen so far, none of this was neural network. This is just using pure thought and physics and some computers. More computers, less thought. We just worked out the equations and do this thing by patch. But then you can now tell the same thing to a neural network. Like you can tell the neural network how the image was formed as it passed through the different parts of the object, and then ask it to refine what you guessed using that pop out. Okay, so quiz number 3. What is this? This is a two-dimensional image of a thing. What is this thing? It's kind of hard to figure out because you probably never thought about this. This is the electron microscopy. It's actually this weird shape. It's all made up, by the way. This is all simulations for now. This is the shape. The question is, can you get the shape? So with pop out, as far as you can push it, you get something that looks kind of like it, but also kind of creepy. Right? Now you give this as a starting structure and you give it a neural network and say, okay, I tell you the propagation of a wave through the object. I know the propagation physics. It's basically Poisson's equation with some absorption, blah, 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 blah. Send that in, refine the volume for me, and I told it the physics. And you get something pretty close. So neural networks can now, these are what we call like physics-informed neural networks. You can put in scattering processes into the layers of a neural network and ask it to refine based on that, matching what you see here with what the structure is there. So it takes this as a starting point and say, what do I have to change here, knowing the physics propagating forward to create an image where it doesn't match well, I change that little thing until it refines well. Okay, so this is the optics part. We use it with butterfly scales. I guess nobody really cares about butterflies here. Butterfly scales are nanophotonics. The reason for the blue color is structural. It's the structural color. It's not a pigment. So if somehow this gets wet and it's a thin film interference, okay? Think about it that way. So the thickness is quite important. And so we've used X-rays to do the same thing, but so these are different sort of scales with these very interesting 3D structures. The important thing is the thickness of the bottom film is tuned to give it a brown appearance or a blue appearance. But to do this is expensive and slow because we do it at synchrotron. It takes a lot of time. But we want to do this with any electron microscope here using this pop-out technique. I might have to, I'm going to skip through this because I think I'm running a bit out of time. It was done by this guy and this guy. One of them is at the back. You might know him. This is partial coherent imaging. So the idea, I'll say a little bit since I'm already here. The idea is that when you try to do coherent imaging, you need to filter the energy to be elastically scattered. But if you do that, you lose a lot of the electron counts. If you know the inelastic scattering processes, then you can also use the inelastically scattered electrons, try to increase your signal to noise. So it's the same sort of stuff that what I sort of talked about, putting the physics into a neural network. except now you also put some of the losses and all the inelastic processes inside. I'll skip this. I'm very sorry. I'm very sorry. Sorry, Joel. Okay, but here's the neural network. The neural network is usually very confusing to look at, even for me. But I want to point out that in the neural network, it takes the input and tries to guess what the output of the object is. There are two parts. There's one part which we stuff all the physics in of the propagation. And then there's a fudge part, which is all the physics that we don't know about. That can you please make it look like the image that we saw? If you did an end-to-end learning, this would be gone. You just use end-to-end, but you lose sight of all the physics. If you know the physics of information, you should put it back in. If you know the elastic losses, you should try to put it in. And what you have to fudge in to figure out the difference between the theory and experiment gap is where the new science could be found. Right? Blah, blah, blah. Anyways, just to show you the results. This is a very noisy little protein in there, cryo-electron microscopy energy filter. Low pass filter, this is what the ground truth is. So that image is of this thing. It has ice and is noise. Really kind of weird and difficult. So with a pin, you can actually get this sort of images out. And that's because we put in as much of the image formation and the inelastic losses in as we can. Okay, back to motif hierarchies. Sorry for the 15 minutes. Okay, yeah. So this is the fun part. Now I'm going to get to some marketing. Means I need to take off my jacket. Not because it's marketing, but because I'm getting warm. So these are other examples of motif hierarchies that I didn't tell you about. Some of them. There's water, there's topological materials. You've seen that one. You've seen this. This is for biodetection. There's some for metal organic frameworks everywhere. There are motif hierarchies in our models and in our science. Okay. But science or ML alone, if you come only from that direction, is insufficient. So we need to create bilinguals. To create language models of complex science, right? So tokens, grammar, and context. Tokens map to motifs, hopefully. I've made a partially convincing case. We need to look for motifs. Then we need to think about how motifs interact, like the grammar. Then we need to generate versions of these and check against our experiments. And that's where emergent phenomena occur. Okay? So how do we do this? How do you do this? How do you get involved? First, you ask ChatGPT to create an image. This is quantum computing in the style of, you can guess what the style is. There's a lot of issues with this problem. So we need these people. These are students, grad students, solving the language of complex systems. These are whoever you are. That's what you look like. You mean quantum point putting. Okay. So we have created an AI for science masters. Okay. I've sort of wanted to do this, but in a larger context or maybe a smaller context, but this is a very nice sort of opportunity to do this. And the way this is crafted is there are some core AI tools, very skinny little bits for pre-deep learning and post-deep learning and deep learning stuff. The thing I like a lot is we get to create a completely new program based on what I think is important or we think is important. So this one is a very nice one, applications of AI in science. I have not really shown you an equation yet, except for the friction and the entropy equation. But I'm able to get you through many of the machine learning concepts just with my hands and my words and fancy graphics, right? I think we need that as a skill. So the applications of AI in science is actually reading papers and explaining it with as little math, as little computation as possible, okay? Core AI. Then there are electives. Electives are basically the different sort of subtopics. This is a growing sort of set. AI and astrophysics, Mark Hahn coming into physics, he'll be teaching that. Bioimaging, that's me. In chemistry, pharmaceutical sciences, we're adding condensed matter physics, AI and condensed matter physics, AI and optics, and whatever it is that people think needs to be taught. But these are different specializations thinking about the different problems. And then, because it's AI in science, if you don't remember or you need some of the science, you should also be able to pick it up for free. So in this master's program, students can also take two to fulfill their graduation requirement, 4 or 5,000 level science, faculty of science course, anywhere within the faculty of science. If you want to do chemistry, take that. So this is how we want to shape the whole thing. So do we have bilinguals? Yes, and it's so the application is closed. I don't think any of you actually qualify. Because that's why you're here because you're not there. But and we have a lot of applicants. It's very crazy. But if you have friends that are interested, please let them know. Okay, so the bigger question is, yes, we have this program, blah, blah, blah. It's all great. We're trying to educate people. What is it that they're trying to do? Well, this is the language analogy. Tokens, motifs, language, complexity. Okay. Unnecessary and okay. So the other thing that the masters will solve, and this is where I need the senior people in the audience for your help. Senior people. If we were to really try to flesh this out, we would try to use machine learning to attack different fields. It's like trying to learn a new language. What's the starting point? You have to learn the grammar. Well, you have to learn the words first. You have to find the tokens, then learn the grammar. And this, for a lot of us, especially people that are soaked in experiments with a lot of data, tokenization is the barrier. If you haven't figured out what are the patterns or what are the words that goes into a language, you can't really start grammarizing or generating stuff from it. Unnecessary. Yes, and bilinguals. Okay, so this is my imagination of the grand scheme of things. Okay. The grand scheme of things, red stuff, is the barrier. It's the tokenized stuff. But if you have data sets that is pretty complex and you've had it around for a while, you don't know what to make of this, we can try very simple tokenization with the unsupervised learning methods first. If they work out and you see tokens, then you can build the grammar and you can build interpretable models, generate hypotheses, and then test it in the lab, right? And then later on, you can go collect your Nobel Prize. So this part, a lot of us can do. Okay, I can do this. If I can do this, undergraduates can do this. Undergraduates and master's students. That's what the master's program, I think, can really give us a boost. It help tokenize a lot of these in the starting stages. Then we need a longer timeline for PhD students to create models. This is usually typically the harder part, because then you have to tweak stuff, you have to compare with experiments, maybe do some experiments, and then really generate hypotheses. And this is a longer timeline that would only work if this is already an available source. Then after that, PhDs and postdocs, this is typically the line. Like if you're expected to write a nice paper about it, you should do the whole thing. If I hire a postdoc coming in, I expect the postdoc to know how to do all of this. Okay? But then this right now, for a lot of us, and this is where I think I can try to engage PIs or different researchers where you have a lot of complex data and you don't really know what to do with this. And we think about this from a language and complexity point of view. If we can make models of this. Then we can do experiments and tests. I don't know what this is. The way to make this happen is not just the masters, it's to create a gym. So the gym, I actually go to the gym. I'm out of shape. I still go. At least it keeps me less out of shape. Okay. So the gym is inspired by this idea. It's a practice place. It's a place where if you have projects and so on and so forth that's done by some junior person in the lab, you want to pass that on to the next person coming in. But how do we do this? We do this random shares. Just share this to that guy and so on and so forth. You talk to this person. Can we have a place where all of these are collected, where there's a pool of data and there's a pool of pedagogical examples that work on data from PIs here, not just cats and dogs and digits from whoever the heck made these things. We don't even care about. It would be nice if one of the students came in and said, "Hi, Professor Valerio, I worked on your data set. Who are you?" Not the student. You would say to the student, "Who are you?" Right? Not, "I know who you are. Sorry." And then you say, "Who are you?" "Well, I'm a student. I'm a fan of your data on the gym. I know how to work on it, and I work with you." That will be a nice win-win. And then if, let's say, the student leaves and then another student comes in, and then Professor Valerio would say, "Well, why do you pick up from where student A was? Continue from there. There's already an example of tokenization. Build models on top of that." This will outlive, hopefully, any single research grant or project that we have. It will be a nice collection based on a growing set of data sets that we can curate within the NUS environment. And hopefully, if we extend out to the Singapore ecosystem. I'm saying this like it's funded. It's not. Okay. The Gantt chart is not funded. But the idea is the first phase is we want to build at least 15 to 20 projects, and we're trying to fill this. We're trying to fill this with meaningful things that we know could work in the tokenization phase for the first year. Then the expansion phases would be starting to model on top of that, right? So we add more projects, but widening the scope. I'm in physics and biology. I talk to physicists, I talk to biologists. Many of the projects will come from these initially. But once this works, we could broaden, and we should, to other different fields. And then the last one is, I don't know, sustainability, something. We get funded. These are the people, the PIs that have already committed. Okay, this is not a shaming exercise. Just kidding. Alex is there. He has 2 projects. Put him at the very top. Also because there are, I think, 25 projects or so. These are already agreed. Now, the interesting thing is if you have a data set and you're still not really sure about machine learning, I don't know, you're not really selling this very well, and you want a co-supervisor, we can find you an ML or an AI co-supervisor. But we can only do it for that many. But if you're bilingual, which means you're a PI that knows both, comfortable with both, I'm happy for you to put something up. It'll be really good if you can contribute a pedagogical piece into the gym. So would you like to join? If you would like to join, please write me. Okay? I think that's kind of it. Okay, thank you for your attention.